in this chapter, I'll be looking at chapter 4, carbon and molecular diversity of life. So I'll be looking at the macromolecules, look at carbohydrates, we look at protein, we look at lipids, we look at nucleic acid. And if we look at all of these macromolecules, you realize that they are predominantly made by carbon. So aside from water molecule, carbon is the most predominant compound element that dominates other compounds that we are going to be talking about. So compounds that have their backbone filled with carbon, we call them organic molecule. And we have 4 main classes of some of the molecules we'll be looking at this chapter. We are looking at carbohydrates, we are looking at lipids, and nucleic acid. Now, we say that the first 3 that you can see are the huge molecules, macromolecules that we are very much conversant about. Then we have the nucleic acid, which I'm going to discuss also to details. What determines an electron, what determines an atom's electronic configuration is a kind of bonds that makes up the element. So we talk about the kind of bond, the kind of molecules and elements that makes it up. We'll talk about some kind of ionic compound, polar compound. So like water now, we say water is a polar molecule. And the reason it's a polar molecule is simply because it is formed by a polar covalent bond, which is a covalent bond that is formed by unequal sharing of electrons, because oxygen have higher affinity for electrons than hydrogen. So we say that oxygen is a high electronegative element. All right. So be looking at all of that. Now, when you say the valence electron, the valence shell, if you remember from the previous chapter, the valence shell is the last shell that makes up, is the outermost shell of an electron. All right. So carbon have 4 valence. The reason we say carbon have 4 valence is that when you look at carbon, it is number 6 on the periodic table. And being number 6 on the periodic table, it has the innermost shell have 2 electrons and the outermost shell have 4 electrons. So with that, there are 4 unpaired electrons in carbon outermost shell. So that means carbon is able to make 4 bonds. It's able to make 4 bonds with other elements. Now, this is what gives carbon the privilege to be able to form larger compound or larger or complex molecule. All right. So when you look at this, here is what I was talking about carbon's ability to be able to make bonds. So when you look at this, for instance, now this is carbon here, and you see the valence of carbon here showing that carbon is able to make 4 bonds. So we say that carbon have a valence of 4. So here you can see that carbon combined with 1 hydrogen here, 1 hydrogen here, 1 hydrogen here, and 1 hydrogen here. So we say this is a saturated compound. All right. Here again, you see carbon here, C2H6. It's a carbon binding with other molecules. Again, what I want to stress here is that carbon can make 4 bonds with each of the carbon molecule. So if you count this now, there are 4 bonds attached to this carbon, and there are also 4 bonds attached to this carbon. Now, when you come here now, we say these compounds are saturated compounds, and these are in the alkane groups. Alkanes is spelled AL, so I'm going to use this. So these are alkanes, methane, and these are alkanes group. Now the alkanes have the formula CnH2n plus 2. And so when you look at this, when C equals 1, so it's C1 H2n plus 2. So that is C1 H2 times 1 plus 2, meaning that gives you CH4. And here the same formula, but when you come here, you say it is CnH2n. So you can see you see carbon, there are 2 carbon here, then 2 times 2. So that gives you C2H2n. This we call this the ethane. All right. So this is unsaturated compared to the first ones, right? Okay, let's go. Now, when you look at this, so looking at this now, the electron configuration of carbon gives its covalent compatibility. Now, don't forget that covalent bond simply involve sharing of bonds, sharing of electron by two elements to form a bond. Now, the bonding can be equal sharing and it could be non-equal sharing. When it's equal sharing, we call it non-covalent, non-polar covalent bonds. When it is equal sharing, we call it polar covalent bonds. I mean, when it is not equal, we call it polar covalent bonds, meaning that it draws to one pole to another. That's what you see in oxygen and hydrogen in forming water molecule. When it is non-polar, that's what you see generally when molecules are formed between two elements. Hydrogen and hydrogen atom is going to give you hydrogen molecule. Oxygen and oxygen atom is going to give you oxygen molecule. All right, so the electron configuration of carbon gives it covalent compatibility with many different elements just because it has valency of 4. Now, valency of carbon and its most frequent partners, which are hydrogen, you see the hydrogen combined with oxygen carbon very well, with oxygen, all right. And with nitrogen, they are the building code that govern the architecture of living molecules. So we're going to be looking at that. Hydrogen have valence of 1, so that means 4 hydrogens are required to relate with 1 carbon. Then oxygen, there are 2, that means 2 carbon, 2 oxygen are required to relate with oxygen. Then we have nitrogen, then we have carbon itself there. All right, so when you look at this, carbon is able to partner with these atoms here. So you see carbon forming bond with oxygen. Then you have CO2. This again is a non-polar covalent. See here. Then here you can see carbon binding with oxygen here. Then it binds with nitrogen here, while nitrogen also binds with two atom of oxygen of hydrogen. Then you see the same thing here. So there are 4 bonds again attached to nitrogen here, to carbon here, you can see. All right, so carbon is the one that makes the scalar thing for most of the organic molecules that are in our system. So when you look at the thing, not just in our body system, but in our system, right? So when you look at the thing, for instance, now, it is C2H6. And if you look at this, you see C2H6 here. This is the backbone of this compound, and it is fully saturated, like I pointed earlier. Here is propane, meaning that there's C3H8. And here you see the presence of double bonds here. This is C4, and you can see this is H. This C4, then H here, meaning that CnH2n. All right, here you see C4 and H8 as well. All right, but what is predominant here is that you can see double bond. You see double bond is on the first carbon here, so it's called 1-butene. Whereas double bond is on the second carbon here, it's called 2-butene. All right, now if the double bond are to be here, then we're going to start counting from here. We are not going to call it. So if double bond shift to this here, then this become the first carbon in that instance. All right. Now here you have butane here, which is CnH2n plus 1. Then you have 2-methyl propane. Now 2-methyl propane, in that you can see this is here. Let me use my notation. So here is the propane, which means 3 carbon compound. Then there is an attachment. This is called methyl. This is called methyl. And the rule is CnH2n minus 1. So you can see that's methyl there, CnH2n minus 1. So the methyl gives you CH3. Now the CH3 is attaching to this carbon on the second position. So that's why it's called 2-methyl propane. Now, if you look at this, you have cyclohexane. Cyclohexane means that it is, if you count the number of carbon, you have 6 carbon, which imply hexane. And then if you look at this, you have it is cyclic, so that makes it cyclohexane, and this is called benzene. All right, let's go. Now, so when we say hydrocarbons, we are talking of hydrogen and carbon molecules. So they are generally carbon molecules that are formed by the combination of hydrogen and carbon. Most organic molecules are actually hydrocarbon molecules. So you can call them hydrocarbon components, like fats, they are hydrocarbon. Like for instance, now, some of those molecules, rather, you see they are not glucose, you see that they are majorly predominantly made up of carbon and hydrogen. So any of the molecules, if you look at your octane that you put in your gas in your car, it's made up of carbon and hydrogen because it's octane and it's just C10H20, which is predominantly hydrogen and carbon. Hydrocarbons can undergo reaction that release large amount of energy, and you can see that in the gas that you use for transportation. Now, what do we call functional group? A functional group is simply the face of the compound. All right. Now, the face of the compound tells you the identity of the compound. It also tells you the function of the compound. You remember in anatomy, the anatomy determines physiology. All right. So it determines the shape of the compound and the identity of the compound and also the function of the compound. That is functional group. So whenever you see OH, for instance, now you can tell that this is hydroxyl group. Anytime you see, and that tells is ethanol, alcohol, that is OH. Anytime you see a compound that is predominantly filled with COOH, you can tell that this is carboxylic group, right? So there are chemicals like that, there are symbols like that that depict the actual identity of a molecule or a compound. That's what we call functional groups. So it's a compound. Functional groups are components of organic molecules that are mostly involved in chemical reactions. So it determines their function, it determines their reactivity. The number and arrangement of functional groups give each molecule its unique property. All right. Now, here are some functional groups that you should be familiar with at this point. You see the hydroxyl group, which I mentioned earlier. Now, so when you look at this, you see the hydroxyl group. Anytime you see OH in a molecule, in a compound, you can tell that that is an ethanol or an alcohol. So the general name is alcohol. An example will be ethanol, methanol, propanol, octanol, pentanol. So all of those molecules, when it ends with when it has the OH anywhere on that compound, it is an ethanol. It is an alcohol. Now, when you see this symbol is a symbol for either ketone or aldehyde. So when you see this here, that is a ketone. When it is at the terminal, that is propanol or aldehyde. Now, this is COOH, which I mentioned earlier. Now, when you see, you can tell that this is carboxylic group. All right. So here is carboxylic group. You can see that it is carboxylic group there. Now, and amino group is right here, NH2. And so that's amine, that is glycine, which is an amino acid. Then SH is sulfhydryl, and you can see thiol there. All right. This is also an amino acid. Now, when you come here, you see phosphate, which is usually part of the, it's also part of nucleic acid. DNA has phosphate, we'll talk about that. All right, then you come here, you see methylated compounds. Simply means any compound that have methyl, which I told you earlier. The formula that produces methyl is CnH2n-1. So you see, when N is 1, it's going to be CnH2n-1. I mean, when N is, it's going to be 2nH2n plus 1 rather. So that gives you CH3. All right, so it's CnH2n, like Cn, let me see if I can write this, H2n plus 1. Right, so that's C1H4, I mean C1H2 plus 1, that makes it H3, so that's CH3. All right, so when you say methylated compound, it means a compound that have methyl. So this is methyl attached to this compound. However, this compound itself is cystone. So the carbon, the methyl is attached to carbon 5. So that makes it 5-methyl ketone. I mean, methyl cytosine rather. That's cytosine that we're going to be talking about when we get to DNA, RNA. All right, so those are the major functional group that you should be aware of at this stage. You should be aware of hydroxyl group, carbonyl, carboxyl, amino, sulfhydryl, sulfophosphate, and methyl. Now, you may not be really talking about sulfhydryl, but you'll be talking about the other ones very well. All right. So when you look at this, for instance, now, when you look at your ATP, which is what we call adenosine triphosphate, the guy that gives you energy. Now, adenosine triphosphate means it has adenine in it. It has adenine or adenine. It has 3-phosphate, which is where you got the name ATP. Now, that is the guy that gives you energy. Anytime you need energy, that is energy currency. Now, adenosine triphosphate is not made in reserve. You don't make it that you're going to use it later. The body makes it as it's needed, right? Now, so it consists of organic molecule that is called adenosine attached to a string of 3-phosphate. So here are the 3-phosphate right here, and this is the adenosine. All right. Now, if you need energy, for instance, what happened is that there are 3-phosphate here, and it is this last phosphate that gives you energy. So this phosphate will just go ahead and join one protein. To just join one protein. When the protein receives the phosphate, your body gets to work. When the phosphate is not there, the protein is inactive, cannot do anything. So that's why when we talk about protein is a machine in your body, ATP gives the power for the machine to work. And you get ATP from carbohydrates. All right. So once you remove the phosphate, how many phosphate is going to be left here? It's going to be 2. And that's what you see here. Now it becomes ADP, adenosine diphosphate. Now, so what do we call polymer? Polymer simply means more than one monomer. All right. So there is something we call the monomer, which means one unit. Then when you build with a monomer, you're going to get a polymer. So monomer is single. When monomer is added together, then they become polymer, which is a large structure. And I'll be talking about that. So how do you join monomers together? If you have one monomer, or if you have 2 monomers and you want to join them together, the process that joins them together is called dehydration reaction. Now, how does that happen? So let's say this is one structure, this is another structure. If you are going to add this to this existing polymer, this is a monomer, 1 unit. If you want to add it to this unit, this will lose water molecule. So you see OH will be detached from here, hydrogen will be detached from here, it forms water molecule. Now water molecule will be removed, then they are going to form a larger molecule. That's why it's called dehydration, removal of water molecule. So dehydration reaction is removal of water molecule to form a bond. Again, removal of water molecule to form a bond. And then the other reverse of that is hydrolysis. If I have this molecule here now, and I want to break it into smaller molecule, what do I have to do? All I need to do is to restore the water molecule that I removed. So addition of water molecule to break a bond is called hydrolysis. Hydrolysis. Addition of water molecule to break a bond. All right, so it's a reverse reaction. Again, dehydration, removal of water molecule to build a bond. Hydrolysis, addition of water molecule to break a bond. All right, so each cell have a lot of macromolecules that are present in them, and the macromolecules are also of different diversities in different organisms, and it's very more within species. And what I also want you to establish is that, again, polymers can be broken down to make monomers. That is what we call hydrolysis. And again, the monomer can be coupled together to form polymer, which is dehydration reaction. I'll be talking about that to detail as we go. Now, so let's pick up each of the macromolecules and discuss them. So we are going to look at, first we are going to pick the carbohydrates. So carbohydrates, what are carbohydrates? Now, carbohydrates are simply generally saccharides. So we call them sugars. Now, the sugars, they can be, so we say that it includes sugar and the polymer of sugars. Now, the simplest carbohydrates are the monosaccharides, which we call single sugar or simple sugars. Now, carbohydrate macromolecules are polysaccharides, polymer of monosaccharide. Does that make sense? All right, let's go. Now, so if you look at this, for instance, now, you see that we have on here, all right, so we have the formula for monosaccharides, which are usually multiple of this. Now, the right way to write this is CH2O. So the 2 is for hydrogen there. All right. So just the same way you have these ones. All right. Now, so glucose is usually written as C6H12 and O6, just like what you see here. All right. Now, so monosaccharides are actually the glucose, fructose, and the last one, which is galactose. So glucose, fructose, and galactose, those are the monosaccharides. Here is a sugar molecule that we call ribose sugar, and I'm still going to talk about that more as we study. Now, if you look at this, if this is a linear form, this is sugar molecule, and this is usually the way you see it most of the time when you are studying, but this is actually the shape. All right, so this sugar molecule is called a ribose sugar. Is a ribose sugar, is a pentose sugar. Pentose means 5-carbon sugar. Now, so when you see this, I'll come back and talk about this later. Now, I don't know, for some of you that may have heard of ribose sugar and deoxyribose sugar before, you will take note that deoxyribose, the difference between deoxyribose is that the H, the oxygen here, is not there. It's already missing that oxygen. That is the kind of glucose sugar that you see in DNA. And I'll talk about that later when I get there. All right? All right, so this is a disaccharide. Again, for monosaccharides, I told you I told you to link it as FGG, fructose, glucose, and galactose, FGG. Now, when you get to disaccharide, it's simply to couple 2 monomers together to form a dimer, to form a disaccharide. So sugar means, saccharide means sugar. Mono means 1. So 1 sugar, that is monosaccharide. So for all monosaccharide, it's going to be, let me get these to type here. So, example, all the monosaccharides are in this category. They are number 1. I'm just going to write here. Number 1, you have the fructose. Number 2, you have the glucose. And number 3, we have the galactose. Those are the 3 monosaccharides. Now, so when you get to disaccharides, the 3 are going to be combining together randomly to form what we call disaccharides. Now, there are 3 disaccharides as well. The first disaccharide is so there are 3 of them. I'll just type it here. So number 1, we have, I use the word LSM, right? So the first one is lactose. And lactose is formed by glucose plus galactose. And number two, we have the sucrose, which is formed by glucose plus glucose. Sorry, let me use this. Let me do maltose first so that it will come up with the LMS. So maltose is glucose and plus glucose. And the last one, which is sucrose, it is glucose plus fructose. Now, what do you notice? You notice glucose is present everywhere. All right. Now, so I use the acronym LMS, like learning management system. So it's lactose, maltose, and sucrose. All right. So those are the element, the disaccharides. Now, what form? The sucrose is your table sugar, right? All right. Now, so this lactose is the lactose that some of us have lactose intolerance of, right? It's because it's a disaccharide, it's not as easy to break down like glucose. Now, there is a bond that formed between them, and here is the bond that you see here. This bond is called a glycosidic bond. Again, this is a kind of dehydration reaction. You see, a water molecule is removed so that a glycosidic bond is formed between two monosaccharides. So removal of water molecule to build a bond is called dehydration reaction. Does that make sense? And that helps to couple monomers together to form polymers. All right. Now the third kind are the polysaccharides. For the polysaccharides, it simply means they are bigger than two monosaccharides joined together. So anything more than two monosaccharides is polysaccharide. So from 3 monosaccharide upward, that will be. Now the polymer of sugars, they have storage and structural roles. The structure and function of polysaccharides are determined by sugar molecules, the monomers that makes it up, and the position of the glycosidic bond that forms them. Here is it. Now, number one, I say that you will see that when you eat glucose, and let's say you ate yesterday night now, and you had a bowl of noodles, by the time you are going to sleep, you have not used up that noodles, right? So if you start to work throughout the day, you are using a residual energy, a residual reserve, a residual resource, a residual energy source that you have stored in your body yesterday, in quote, over the time. What is it? When you eat glucose and it is not used, then it is stored as glycogen. All right. So when you use glucose and you don't use it, when you eat glucose and you don't use it, your body automatically stores it as glycogen. Your body does not store it as glucose and not starch. So when you eat starch, you eat any food source like that, your body is going to store it as glucose. Now, so is the storage polysaccharide in animal? You see that? That is how polysaccharides are stored in animal. That's why glucose are stored in animal. All right. Human and other vertebrates store glycogen mainly in liver and muscle cell. And when it is time to use it, your body is going to go back there and break up that glycogen to make glucose from it. Does that make sense? Yeah. So now, for plants, since you have a way of storing glucose, plants also have a way of storing glucose. As a matter of fact, that's why you are able to eat potato. That's how you're able to eat rice. That's why you're able to eat spaghetti. All of those food are starch sources. They are starch. All of those starch sources of food, they are simply produced from plants. How plants stores glucose. And so plants store glucose as starch. Again, you don't store glucose as starch because you don't produce food. All right. So plants store glucose as starch. And you see plant surplus starch as granules. The simplest form of starch is the amylose, which is what is digested by the enzyme amylase. All right, now the third one is the cellulose. Now you will notice that plant cell have cell membrane. I mean, they have the cell membrane, yet they also have the cell wall as an additional protection. That is why plant cell don't burst. And so you cannot try plant cell if you put your blood cell, your red blood cell in hypotonic environment and put plant cell in hypotonic environment. Plant will survive the environment, whereas your cell is going to burst because your blood cell does not have central vacuole which help them to contain water, so they are going to be immobilized. Whereas plant cell, even though the turgidity could increase, but they are never going to break. Why? They have the central vacuole. Number two, they have the cell wall that protects them, because cellulose is a polysaccharide and it's very tough to break. Now, so polysaccharide cellulose is a major component of the tough wall of plant cell. You can see that. And then you have like starch and glycogen. Cellulose is a polymer of glucose. You see that. But the glycosidic linkage in cellulose differ. The difference is based on the two ring forms for glucose. All right, let's go. Now, in starch, the glucose monomer are arranged in alpha conformation. Then starch, and glycogen, that are usually largely helical. All right. Now, in cellulose, the monomers are arranged in beta conformation, and cellulose molecules are relatively straight. All right. Now, I will. Okay, yeah, these are things. So chitin is also another way that polysaccharide also appear. So polysaccharide appear as starch, glycogen, cellulose, and chitin, which is what you see. Some people call it chitin, or chitin. All right. Now, this is what you see in skeletal system of arthropods. Arthropods have this chitin or chitin as their skeletal structure, because they have their skeletal system outside, right? Now, so it provides structural support for the cell wall of many fungi as well. So I'll talk about lipids in the next video. Thank you.